there. Welcome back to Dr. K. Primer lecture series presenting by Dr. K. Prem. That's me. Today we will talk about the restriction digestion and how the restriction digestion takes place. Before going to the restriction digestion, let us learn about restriction endonucleases. Restriction endonucleases are the enzymes which catalyzes the breakdown of phosphodiester bond of duplex DNA at specific recognition sequences. I'm telling you again, restriction endonucleases are enzymes which catalyzes the breakdown of phosphodiester bond of duplex DNA at specific recognition sites. Right? Let us see the same thing with this cartoon. So here, this is a restriction enzyme which is recognizing the specific recognition sequence and catalyzes the breakdown of this site only, where a first break between the two nucleotides of one strand and second break is opposite side of the complementary strand releases the DNA fragments with the cohesive or blunt ends. So this kind of pattern, this kind of cleavage gives the cohesive ends. As I told you that restriction endonucleases are the enzymes which catalyzes the breakdown of phosphodiester bond of duplex DNA at specific recognition sequences and releases the DNA fragments with cohesive ends or blunt ends. And these recognition sequences are palindromic in nature. Recognition sequence or specific recognition sequences are palindromic in nature. You see, GAA-TTC. If you read from this direction, you see GAA-TTC and you see GAA-TTC. So if you read from right to left or left to right, you get the same sound, right? And the digestion of the duplex DNA by restriction endonuclease have 3 steps. The first one is scanning, recognition of specific recognition sequence, and the cleavage of the site. So when you digest the DNA with the restriction endonucleases first scan the given DNA, duplex DNA. For what? They'll identify the recognition sequence and then followed by the breakdown of phosphodiester bond within the restriction site. We'll see the same thing here. So enzyme is here and it's going to be scan the target sequence and it will identify the specific recognition sequence which is a palindromic in nature. GAA-TTC is a EcoRI restriction site, and then followed by it breaks down the phosphodiester bond within the restriction site. So this is all about the restriction endonucleases. And the restriction digestion. So we will see the sequence of events happens in the restriction digestion in the next slide. So as I told you that restriction endonucleases scans the template given DNA molecule, duplex DNA, and they'll identify the specific recognition sequence and they'll break down the duplex DNA at specific recognition sequence, right? Then they give the DNA fragments, right? When you take a linear DNA molecule with the 2 restriction sites, so here this linear DNA duplex DNA molecule is having 2 restriction sites, EcoRI on restriction site, GAA-TTC and GAA-TTC. When you break down, when you digest with the EcoRI enzyme, it can give 3 DNA fragments. When a linear DNA molecule is having 2 restriction site, it can give 3 DNA fragments, 2 plus 1, right? Then you can see the same thing right now. You see, scanning, and then followed by this. First step is scanning. The second step is identification of recognition sequence, then and the breakdown or cleavage. So scanning, recognition, and cleavage of a restriction site. Again, with the one more molecule of restriction enzyme, restriction endonuclease, you see, it is scanning the target DNA molecule and identifying the restriction site, then followed by cleavage of restriction site. So when a linear DNA molecule is having 2 restriction sites, if you digest with the appropriate restriction endonuclease, then it will give the 3 DNA fragments. 3 DNA fragments. One, which is corresponds to the first restriction site. This is the middle part, and the third one, right? After digestion, if you run the digestion mixture on the agarose gel electrophoresis, which will separate the DNA fragments based upon the molecular weight of the DNA fragments, then this digestion can give 3 different fragments, 3 DNA fragments on the gel. So the DNA fragments resolution on or separation and DNA agarose gel electrophoresis based on the molecular weight of the DNA fragment. So the higher molecular weight DNA fragments runs slowly. The lower molecular weight DNA fragments runs fast or speed. So then the DNA fragment with the higher molecular weight stays at the near well, and the lower molecular weight DNA fragments will be at the bottom of the gel. So when you do the agarose gel electrophoresis, so these DNA fragments arrange based upon the molecular weight of that fragment. You see, this is the bigger fragment and stays near to the well where you load, and followed by the next big one, and followed by the smallest one. The same thing you can see in the agarose gel electrophoresis. If you look at the sizes of these 3 DNA fragments, they are the first one, that is 42 bases. Because of the large size, it will run slowly and it stays at the where you load it. Then followed by the next big one is 34 bases. The next big one is the 30 base pairs. So these DNA fragments are separated on the agarose gel electrophoresis based upon the molecular weight, and we can recognize the number of DNA fragments formed after restriction digestion. Right? Hope you understand the concept. In the next slide, we will see how you constitute the restriction digestion reaction. So always restriction digestion reaction is constituted in a 50 microliter volume, not less than the 50 microliter volume. So first constituent is a 110 microliters of target DNA, or that is equivalent to the 1 microgram. Then followed by reaction buffer. The buffer provides the necessary environment as well as the cofactor for enzyme. So the type 2 restriction endonucleases are working as of molecular cloning. So type 2 restriction endonuclease requires the only magnesium. So that magnesium is present in the reaction buffer. So 5 microliters of reaction buffer is added. Then followed by enzyme, restriction enzyme. So we are taking the tenfold excess so that it will cut the every restriction site present in the given target DNA duplex. Then, so we are constituting for 50 microliters. So you look at this 10 microliters and 5, it will become 15, 15 plus 1, 16. And rest of volume can be filled with the distilled water that is equivalent to the 34 microliters of water. So once you constitute this reaction, you can go for incubation at 37 degrees centigrade for 1 hour. So once you keep this reaction at the 37 for 1 hour, then you can stop the reaction by inactivating the enzyme by keeping it 65 degrees centigrade for 5 minutes. Then the reaction is stopped. Then later on, you can separate the reaction on the agarose electrophoresis. You can check the number of DNA fragments formed after digestion. Right. So this is all about restriction digestion. What are the restriction endonucleases and restriction digestion and typical restriction digestion reaction? So hope you like it. If you like it, and I'm thanking you all for watching the video and learning. And if at all you like it, try to subscribe my channel. That's PREM-PRIMER, Dr. PREM-PRIMER. So if someone is in need of this lecture, you can try to forward if you feel it and like it, and write to me if you have any doubts over this lecture or any lecture in my channel. Thank you all. See you again with one more video. Until then, bye bye.